Welcome to EPG Pathsala, and the second module is Understanding the Architecture of Gene and then Gene to Genome. So my talk will be divided into these parts. First I will explain about the gene, then DNA and cellular organization, third prokaryotic and eukaryotic gene, genetic code per se, and human genome, and finally the summary. So how DNA is transmitting information to progeny and its existence is known through the Mendelian law of inheritance. Basically, DNA is a storage place where all the information is collectively stored in the form of different bases. So there are three processes which I have explained. That it first start replication, where the DNA molecule is replicate and perpetuate all the necessary information so that it can transfer to the progeny. Second is the transcription, where DNA is converted to the RNA, and finally a translation where the RNA is converted to protein. So how this happens, here I will explain with the help of genetic code and finally the Human Genome Project. So let's see the composition of life. Here you can see the different life, but the peculiarity or the beauty of these lives are the composition is similar, that in all the biological system the composition is molecular composition is same. Similar organization principles are used at different cellular level. So organization as well as the molecular characteristics or the molecule per se is similar in all form of life. Here you can see the cellular organization where a cell is there and it has cytoplasm, cell membrane, mitochondria, nucleus, DNA, endoplasmic reticulum, lysosome, ribosome, and Golgi apparatus. So these are the basic component of life, but in case of prokaryotes, there is no nucleus. As you know, prokaryotes is the ancient bacteria or archaebacteria or any bacteria that has devoid of nucleus, whereas in the multicellular organism or in the higher cellular organization, which is eukaryotic, there is a nucleus. So basically, if you want to define eukaryotes versus prokaryotes, you can clearly say that eukaryotes are the cellular nucleus organization, whereas nucleus is devoid in the prokaryotes. So DNA is the molecule of life where genetic material is located in the nucleus. The genetic information is stored in deoxyribonucleic nucleic acid, which is the DNA, and DNA contains the information which is required to build an individual. Here you can see in case of human, trillions of cells are present where 46 chromosomes are present, and the length of DNA, if you can measure, it is approximately 2 meters of DNA, which contain 3 billion DNA subunits, that is ATCG base pair, and approximately 30,000 genes are coding for the protein, which you can say the functional proteins. Now we can say what would be the requirement of DNA. As we have discussed, DNA is a key component which transfer DNA from DNA to RNA in the case of transcription, and RNA molecules work as a messenger, and that is the reason why we have taken mRNA for the translation, and protein are the biological workers, because every action has been governed by the protein. Information of the DNA is copied to a RNA molecule and it is called transcription, where RNA directs the protein synthesis in a translation. And the protein 3D structure is determines its function. And information transfer only in one direction, that is, which means it's a unidirectional and it's an irreversible process. So starting from replication to transcription to translation, and DNA is multiplied to 2 copies, transcribed to mRNA, translated to protein, and function is carry on. Let's see the structure of DNA is a polymer of nucleotide monomers, has 2 deoxyribose sugar and 4 bases A, G, T, C, and here you can see the aromatic carbon rings, and phosphate as well as the sugar. Now come to the purine and pyrimidine, which is the key component, and it's a aromatic carbon structure which preparing the adenine, thymine, cytosine and guanine. So purine base is comprised of adenine and guanine, which is the 2 carbon rings, which you can see here in the adenine, which is depicted as A, and guanine is G in the left panel, while pyrimidine bases are thymine and cytosine, which has a single carbon atom, single carbon ring, and you can see here the thymine and cytosine. Now see the DNA chain. In this DNA chain, you can clearly see the phosphate, sugar and base. So in the right hand side, it's a sugar, in the middle, it's a phosphate, in the left hand side, it's a base. So nucleotides are joined with phosphodiester bonds, and sequence of bases vary from one gene to another gene in order to carry out different functionality or different genetic information. It is extremely long chains which after translation give you desired or any number of protein. Come to the double helix. You can see the base pairing between the thymine and adenine and guanine to cytosine. So 2 polynucleotide chains are joined, double helix twisted in right-handed way, and the full circle of one twist is 10 in every 10 base pair. It's a ladder-like structure. Bases are steps. Sugar and phosphates are supporting pillars, and 2 nucleotide chains run in opposite direction, and the chemical direction of synthesis, whether it's a replication or transcription, is 5' to 3'. Now come to the complementarity issues, and the complementarity issue is base pairing, which is the space between the chain is limited purine with 2 carbon rings, pair only with single ring pyrimidine, which is A to T. So in adenine, you will have the 2 carbon rings, while in thymine, it's a 1 carbon ring. Likewise, guanine has 2 carbon rings and cytosine has 1 carbon ring. Interaction is stabilized by the hydrogen bonds, which is non-covalent interaction and dipole-dipole interaction, and the complementarity pairing is vital for the use and storage of different genetic information. You can see in the right-hand panel that how the sugar phosphate backbone and along with the sugar in the 5' region to 3' region is arranged. Now I start with the gene structure or gene architecture of a prokaryotes. Here it's mentioned in A, B, C, D, E, F, G and H form. So start from the F. Basically F is the start site, which means coding from methionine AUG, it is start from F. Before F is the 5' flanking region, and you can see A is the transcription factor binding element site, B is the promoter sequence approximately at -35 base pair sequence away from the start site, C is the -10 base pair sequence, D is the start of transcription, initiation start from purine, E is transcription binding site, which is also called as a Sine-Dalgarno sequence, which is 10 base pair upstream of start codon, that is the AUG. F is the open reading frame where the start codon start making the methionine. G is the final sort of that gene, which is the next in frame, and the stop codon occur at the G, and G to H is separated by the intercistronic sequence followed by a termination signal. So if you can see here, it's in the linear direction, and start codon before the start codon is the 5' flanking region, which has many regulatory elements, while after G is a termination sequence which can stop the process. Now come to the prokaryotic gene in a vivid way, that you have a promoter, cistron 1, 2 and 3, and n number of cistrons can be, and a terminator. So as I discussed that in prokaryotes, due to nucleus is absent, the transcription and translation processes are coupled, and here you can see that 5' to 3' mRNA is then forming the polypeptide in the lead, and where the ribosome is bind to the leading sequence of mRNA, whereas transcription processes also begin. So this is a simultaneous process and coupled process. To summarize this prokaryotic gene expression, the basic mechanism of transcription and translation are similar in eukaryotes and prokaryotes. However, due to the lacking of nuclei, transcription and translation processes are coupled here, and ribosome attached to the leading end of the mRNA molecule while transcription is still in progress. And you can see it in this diagram that how this translation process achieved in the prokaryotes. Come to the eukaryote gene expression. As we all know that eukaryote has a nucleus, so in eukaryotic cells almost all transcription occur in the nucleus and translation occur mainly in the cytoplasm with the help of ribosome. In addition to that, before the primary transcripts form, it can cleave from the nucleus and translocate to the cytoplasm during RNA processing before the production of the mRNA, and it transported to the cytoplasm because the polypeptide chains are also fall into the cytoplasm itself. In this process, there is a process, in this process, a splicing occurs and all the introns are removed. I will explain in the next slide where I will show you the eukaryotic gene expression. Unlike prokaryotes, eukaryotes have exon followed by introns and exon. Likewise, exon, between the two exons, you have one intron. So what is happening during the mRNA synthesis, these introns are spliced out and then it gives you the coding sequence, which is the joining of different exons. Here you can see that start codon, before the start codon there is a flanking region which is also called the 5' flanking regions. In the 5' flanking regions, you have a promoter element. Promoter can be shorter, promoter can be very long. It can be 500 base pair, minus 500 base pair long to maybe minus 10 kilo base pair long. So you can see here that before start codon, there is a transcription initiation site, which is also called the transcription start site, followed by Tata box and cat box, and there you will find many GC rich sequences in the 5' flanking region along with different transcription factor binding element. Apart from these binding elements, you will have lot of regulatory elements, as you can see in the 5' flanking region, that it regulates the gene expression. It has also enhancer, silencer, and the beauty of enhancer and silencer can regulate gene expression. Either it can accelerate or completely terminate, and the silencer and repressor or enhancer can be 10 kilo base pair upstream or 10 kilo base pair of downstream. Now come to the coding part, where you can see that after start codon, exon 1, exon 2 and exon 3. In between these three exons, you will find intron 1 and intron 2, which will be spliced out in the process of transcription, and after transcription, the introns will be spliced out and will give you a full coding sequence or mRNA stretch. So a gene has exon, intron, different cis regulatory element and many silencer or enhancer, and GC sequence, cat box and Tata box. Whereas in the coding region, it will be a start codon as well as the poly A signal where the termination will occur. So the revise the central dogma of life. The revision of central dogma is saying, first of all, accession of the genome, where the gene will be transcribed to mRNA, and this RNA synthesis will process further in order to give you a assembly of the transcription initiation complex, and then it will be provide you protein synthesis with the help of translation, and this protein synthesis will give rise to a protein which has to be processed again, folding will occur, and finally the protein will be degraded if it will be not properly folded. And all the star marks is the control points or the checkpoints where these processes have been tightly controlled and regulated. Now come to the genetic code, because we have already studied about the transcription as well as translation, but how it occurs, as I explained you, with the help of anticodon of tRNA and mRNA, it reads the three letters that is called the codon, and codons are triplet. And it describes how nucleotide sequences is converted to the protein sequence, and unit of the three nucleotide is called codon, and a codon codes for a specific amino acid which is a structural component of a protein. Here you can see the table of the genetic code, where in the left hand you can see the first letter, and the middle top you can see the second letter, and the third is, and the right hand side will the third letter. And in composition, in the combination of these three letters, the genetic code is deciphered, and the 4 base pair can form 64 different codons, approximately 20 amino acids are found in the nature, and these are the regulatory codon. If you can check the codon table, you can find that codons are not only repetitive, it's a degenerative, it's ambiguous, and it can code for many proteins simultaneously. Furthermore, I want to discuss about the open reading frame, which is also an important part in the gene architecture, because if a protein fragment is produced after translation, it has to be in a proper frame. Basically, there are 6 open reading frames, 3 in the sense strand and 3 on the antisense strand. And here I have taken part of the arbitrary sequence, and after translation, the annotated product is starting from methionine. And you can see in the frame 1, there is no stop codon in between. Whereas in the frame 2, after 6-7 amino acid, there will be the stop codon, and similar in the frame 3. So out of these 3 frames, if you have to choose, you will choose only frame 1 because you need a full length protein product and not a truncated product. Because in the frame 2 and frame 3, you have truncated protein product and also not in frame, which means suppose if you will use these protein, it will not expressive enough, or sometime it will not express in your system. So this is the famous saying by the Francis Crick. The meaning of this observation is unclear, but it raises the unfortunate possibility of ambiguous triplets, that is, triplets which may code more than one amino acid. However, one would certainly expect such triplets to be in a minority. So now you understand the efficacy of codons, how it code. You have seen the genetic table, codon table, where you have seen how codons are making many proteins. It's ambiguous, degenerative, repetitive. Come to the human genome part, and this is the here you can see that we all human beings have 46 chromosomes normally, where 22 autosome pairs and 2 sex chromosome pairs. 3 billion base pairs in the haploid genome, whereas there are about 30 to 40,000 genes are functional genes. And now we have to understand, is there anything other point which is important for the study of human genome? The different type of sequence that make the total DNA of a human cell. Suppose if somebody will say that define the genome. The ancient definition of genome is haploid set of a gene in a eukaryotes or in a multicellular level, but now it's bit changed, and the genome is now defined as the entire set of gene present in a chromosome or present in a cell, which means it has the proper gene, proper DNA, it has the junk DNA, it has the regulatory DNA, it has the repetitive DNA. So the complete pool of DNA is now called a genome. So you can see we do have 3 billion base pair of nucleotide sequence, about 30,000 genes are functional, and only 2% of the DNA, if you can compare the ratio between the 3 billion to 30,000 genes, only 2% of the DNA is encoding proteins. Genes are included with the exon, introns, and many regulatory components. Beside coding areas, there are also additional sequences formed, which is in the form of junk DNA, repetitive DNA, and many other VNTR, which is the very long tandem repeat sequence or short tandem repeat sequences. So human chromosome, this is the chromosomal picture. You can see that we do have 23 pairs of chromosome, 44 autosomes and 2 sex chromosomes, and the division with the 2 sex chromosomes in X and Y, where the XX denote female, XY denote male. So this is the picture of the chromosomes carrying the same genes called homologous, and here you can see so how the different proteins are produced, how we are finding a gene mutation in the disease. And here you can see any kind of alteration if occur in the DNA stretch, it will lead to the mutation. Some are the normal mutation, some are the big mutations. It caused due to the chemical changes or physiological changes or error, any kind of error during the process of DNA replication. However, cell has machinery to repair some mistakes if there. If not repaired changes in the DNA sequence, it accumulates and made permanent problem inside the DNA, and it also gives you a disturbed or altered replicated DNA molecule. Now come to the point mutation. You can see the normal DNA, in one DNA strand, you can see the normal bases like CAT, CAT, CAT. However, if there will be a point mutation, one nucleotide will be exchanged. And you can see here in the single base change, everything is similar except instead of A, there is a C. So this single base mutation can cause missense mutation, leads to an amino acid change, silent mutation which does not change the amino acid but it gives you the different effect, and the nonsense mutation cause premature or stop codon. If you recall the previous slide where I have shown you the open reading frame, even any stop codon can disturb the protein production, that is the translation product, and you won't have the proper translated product. This is the frame shift mutation where suppose if the polypeptide or if the translated product is completely formed in the frame 1, by any means of mutation, it will be deleted or added. It can give the complete or full-length protein product in the different frame. So frame shift mutation is occurred either due to insertion or deletion, duplication, translocation, and this results in the altered reading frame and severe impact on the protein structure. So before conclude my talk, I would like to explain something about the genomics per se, that how genomics is important in the molecular biology, because genomics is dealing with the genome structure and function, and with the help of bioinformatic tools one can analyze an enormous amount of data to detect various issues such as variations in the genetic makeup that affect health, what is the cause of a disease, and what would be the drug response on a particular disease. In the gene and genome topic, we also would like to give some glimpses of the genomics, such as the functional genomics, which is the understanding the complex mechanism and relationship between genotype and phenotype on a genome-wide scale. This would help us to understand the DNA structure and sequence, to focus on the dynamic aspect of the interaction, including gene transcription, translation, and protein-protein interaction. This is the functionality part of the genome, and thus what would be the outcome of this functional genomics, and the outcome is as follows, that it can help to answer many questions which is relevant to the biological problems, such as temporal and localized gene expression, in which, where and when these genes will be expressed. Second, gene expression pattern at different cellular level and states, which means how gene expression levels differ in different cells, different cell types, and state also. The third crucial important point is what are the functional roles of different genes and in what cellular processes do they participate. Fourth one is how genes are regulated or modulated. Fifth one is how gene promoter is regulating gene expression in a particular cell type. 6. How do genes and gene product interact? And finally, how gene expression is altered in disease and usage in the therapeutics. As I have already explained you the primitive structure and the advanced structure of DNA double helix, which has carrying the purine and pyrimidine base, what are the different requirements of the DNA, how a DNA chain is elongated, what is the functionality of the DNA double helix, why complementarity is an important issue, and gene structure of prokaryote as well as in the eukaryote, and how these genes are expressed, what are the different prokaryotic gene expression mechanisms, what are the different eukaryotic gene expression mechanisms. We have also touched little bit about the central dogma, which has been revised many times, and the concept of new central dogma theory, what are the usages of the genetic code, how open reading frame is important in terms of study gene architecture, the human genome project, the chromosomes, and what are the different mutations we have discussed already, point mutations, and how point mutations are important to check and to understand the etiology of a disease, the frame shift mutation, how an addition, deletion, insertion, or duplication can greatly affect the open reading frame and thus the pattern of expression. We have already discussed these things. Now come to the common aspects that how these things can be identified, and here the common experimental approaches for functional genomics that enables us to understand the etiology and the functionality of many drugs and disease per se. A common experimental approaches for functional genomics and their enabling technologies are included transfection, and in transfection what we are doing, we are introducing a foreign DNA with the help of certain mediators such as the liposome or calcium chloride or magnetofection. There are many types through which a foreign gene can be inserted into a host system. Then real-time PCR, in this real-time PCR we are checking the production of an amplicon in each cycle, and this will also quantify the DNA amount or DNA concentration using fluorescent reporter molecules, for example cybergreen. Third is the RNA interference, and the RNA interference is an endogenous biological post-transcriptional process through which RNA molecules can be turned off or silent the transcription or translation by regulating targeted mRNA molecules. Then mutational analysis, as I have already discussed this, that it can occur in the living organism during different stages of lifespan. There are multiple factors which is mainly responsible for mutation, such as breakdown in the DNA repair, biological exposure, genetic and natural factors such as chemical carcinogens and UV light. Next one is the SNP analysis, which is the single nucleotide polymorphism, and this is also known as SNP, or the common type of genetic variation in a population. The position in the genome where one individual has only one nucleotide and other has different nucleotide. There are huge number of SNPs in every genome, and a number of SNPs also give rise to restriction fragment length polymorphism. The next one is the microanalysis. In the microanalysis, we are determining the genes are being expressed or not expressed, and this technique also helps analyzing large number of genes at one stretch, which has either been recorded previously or from the new samples. Now come to the important part in the genomics, that is the epigenetic modification, which is another wing of functional genomics. And here I can explain that epigenetic markers include DNA methylation, which is modifying the histone tails, and why methylation is very important in the DNA modification. The main component in chromatin is histones, which are subject to several different covalent modifications such as methylation, acetylation, phosphorylation, ubiquitination, sumolation, where acetylation is the most commonly studied of three modifications and a strong correlation between histone acetylation and active transcription. Why I am saying all this in genome and genomics? Because these things are very important to understand the gene in a genome level. There are many common techniques through which you can identify DNA methylation and histone modification. Number one is bisulfite sequencing. Second is chromatin immunoprecipitation assay. There are now advanced techniques through which you can identify the chromatin immunoprecipitation on chip, which is also called the chip on chip. Additionally, these all techniques used to study DNA modification, including chip, methylated DNA, immunoprecipitation, high resolution melts, and chromatin accessibility assay and microanalysis. There is one pertinent part in the gene and genome is pathway analysis to check the efficacy of a gene expression, and here what we are trying to investigate that suppose biological pathway that include metabolic, regulatory and signaling pathway, how these regulatory elements often interact with one another and to form a biological network. Pathway analysis is used in system biology to build a better picture of how the individual components of a biological system interacts to create the large functional system. In order to achieve, there are many techniques available such as Luminex bead-based analysis, microanalysis, real-time PCR, and proteomics tools such as 2D polyacrylamide gel electrophoresis, is-to hybrid study are the most common. So what would be the outcome of functional genomics to understand gene and genome is, first of all, the production or the discovery of different biomarkers, which is biomarkers can be discovered through differential gene expression, which is identification of mRNA, microRNA or protein expression level as influenced by disease state, therapy or other differences between sample cohorts. To summarize, information passed on in the sexual reproduction needed for new characteristics to develop, offspring receive genes by inheriting chromosome. Thanks for your attention.